get started? All right. So I'm David Shea. So today is Martin's defense. So this is standard in EE, which is the advisor will say a few words to embarrass the candidate a little bit, to throw him off before his talk. So it's been a great pleasure advising Martin. So if I have to think of one word to describe this guy, I would use courage. And here's the reason why. So how did we get into this topic?
That's supposed to be embarrassing?
So here's why. Yeah, positive words are embarrassing. No? I get embarrassed when people say positive words about me, so I thought so when he came to our group, at that time I was really interested in one thing called the James Stein paradox. Really intriguing. And for me as an information theorist with no background in statistics, I decided to learn more about this empirical base and multiple testing. And Martin was courageous enough to come along with me for the ride. So we started reading Brad Efron's book, Large Scale Inference, together. And one thing leads to another, and it became his PhD thesis. Basically applying empirical base and multiple testing in many different problems. So courage is one thing. Second thing is that in the process of doing this research, something very good happened, is that we started working with James Zhang. And James and we collaborate a lot in this project, and that was a good opportunity to work together. And he effectively became informally the co-advisor for Martin. So I'll let you say a few words to embarrass him as well. You want to? This is not embarrassing. You have a very high standard here. I'll think of something for the next student.
I'll try my best. So it's been a really great pleasure to work with Martin very closely for the last 2-3 years. So Martin, I think, is a really very talented student. I think all of you know that. What really impressed me the most about Martin, in addition to courage, is that he's actually really able to solve, I think, really important high-impact problems by bringing in techniques and approaches from different communities. Right, so much of his research is really motivated by really challenging problems coming from genomics. Right? So because of this deep understanding and knowledge and interest in biology and genomics, he's actually able to formulate new problems. And then he's able to apply approaches from machine learning statistics and really push that to the extreme, right? Really make innovations in the algorithms where he's able to prove lots of interesting guarantees, sort of matching lower upper bounds on these algorithms. But in parallel, in addition to just proving things about the algorithms, he's able to actually really develop beautiful, user-friendly softwares that I think will have high impact in the application communities. Right? He's really the complete package.
Oh my god, that was so embarrassing. I used to be quite immature, so
on top of all of that, Martin also tells me that he's actually a very good tennis player. I've read his other works, I've verified his other works. I have not had the pleasure to personally verify that statement, his ability in tennis, but I'm sure with his athletic and intellectual prowess that he will have, his future prospects are indeed very secure. So we're looking forward to your talk.
Great, thanks guys for the very embarrassing introduction. I play tennis a lot, and tennis go and ping me. So thanks for coming to my defense. I'm Zhang. I work with James and David on computational biology. So today I'll start by talking about the data that we have so far. So you know that it is promising that our computers are getting exponentially faster every year, and that is Moore's law, which is shown here in the. Sorry, it died.
It just doesn't work on the monitor.
You can use your finger.
Oh, I can use my finger. Showing as the little blue curve here. So whenever we have data sets that are too big to process, it is always very easy for us to assume that the future computers will be fast enough. But this is not the case for modern data science, and especially in the field of computational biology. I'm showing the orange curve here. The genomic data is growing exponentially faster than the growth of the computing power. which says that in the future, we won't be able to have fast enough computers. Well, the only way to address this, in my opinion, is to develop fundamentally better algorithms. And so today I'm going to address this issue of limited computing power with two pieces of our recent work. And the first work is on, excuse me, on computing the middle for k-middle clustering, and the second part is for accelerating the standard multiple hypothesis testing pipeline that uses Monte Carlo test. And so the first part is relatively easy to understand, and both parts share a very similar idea. So I will briefly go through the first part to build up enough intuitions for you guys, and then we'll hopefully dive deeper into the second part of the talk. All right, so let's start. Oh, and one more thing is that the slides for the first part is really beautiful, and this is modified from some past slides that I made with my colleagues, Vivek and Govinda and Vasilis and my advisor David, and special thanks for them. Well, here's the starting point of the story. So in the year of 2017, we were very excited that we were notified that there was a data set, a huge single-cell RNA-seq data set that was released from this company called 10x Genomics. And so this is still by far the largest data sets in single-cell RNA-seq. And the single-cell RNA-seq is a technology that measures the gene expression level for every individual cell. I know that cells are the basic units of biological systems, and the cells manifest themselves by expressing the genes. So if we can measure the gene expression level for the cells, we can investigate the cell behaviors in a completely data-driven way. Well, so this is exciting. We have this data set, and this is so up to date, and we would like to analyze that. But one thing to keep in mind is that when you work on biology or computational biology, you should always think about the size of the data. And here is the size of the data here. So it has 1.3 million cells, or 1.3 million samples. And for each of cell or sample, it has the gene expression level, which is a number for every gene, for 28,000 genes, or 28,000 features. So it's a huge matrix, and if this is a dense matrix, you cannot even load it into RAM. So you need to think a lot of how to process it in a computationally efficient way. Well, right, so here's the challenge here. Well, so for a data analyst for single-cell RNA-seq data, the first thing they will do when they receive the data is to do clustering. And this is because that for single-cell data is unsupervised. You don't have the cell labels. So you need to do clustering to isolate different cell populations, and then you can identify the cell types here. And here is a toy example of 3 cell populations, and one important subroutine for clustering is to identify the representative cells for every cluster. And there are, for example, k-min uses this subroutine to run its algorithm. Let's zoom in a little bit further to one cell population here. Well, so there are different definitions for the representative point or representative cells. And today we're going to talk specifically one definition, which is the myeloid. And this definition, to put it in English, is that you have n cells, and you want to find one whose average distance is closest to all other cells. And to put it in mathematics, it basically says that we have x1 to xn endpoints here. And I want to find a point y among these endpoints, and whose average distance, or the mean distance, is smallest to all other points. And for the first part of the talk, I will be focusing on how to compute the myeloid in the fastest possible way. Well, first is let's say n is the number of samples and d is the dimension. Then what is the naive method for computing the myeloid? Well, for the naive method, you need this distance matrix, which is n by n, where each entry here measures the distance between point I and point j. And to compute the mean distance for a point here, which is basically the average of a column. For example, this is the mean distance for the first point. And you do it for every point. And then you pick up the guy with the smallest mean distance, that is the midpoint. So that takes n squared time. And with the scale of the data I just mentioned earlier, which has 1.3 million cells, then each single cluster there is usually can be as big as 100,000. So this number is kind of too big and too expensive to afford. So the next question would be, can we make it faster? Well, one thing to realize here is that there are a lot of duplicated computations. For example, I know if point A is close to point B and point B is close to point C, then point A is also close to point C. And by utilizing the triangle inequality, you can, this is not our work, but you can bring it actually below quadratic. But the problem here is that this thing, its dependency on the dimension is exponential. So, and we are in a high dimensional regime, which is pretty bad. So we would like to ask the question, can we make it even faster? Okay? So before introducing the main idea, here is a bit of setup. So here is my nice distance matrix, 8 by 8. I have 8 points here. And so on the right here, I'm plotting the mean distance for every point. So, so this guy is for the first point, and the same for others. Just a sanity check, a question for the audiences. Which guy is the middle? Give me a number. Give me a number here. 4. Awesome. So great. Since everybody understands. Right, great. So this guy is the middle. So now we can talk about the main idea. So there are 2 tricks here for the algorithm acceleration. The first thing, let's focus on the first point here, okay? And this guy is the average of these 8 entries here. Well, the first thing to realize here is that if we do not want to exactly compute it, but instead of that, we want to estimate that, what I can do is that I can take a random subset of samples from this all distance samples. And then, based on that, I can derive a upper and lower confidence bound for that thing. And so the number of samples that I use determines the accuracy for my estimation. So first thing, instead of computing the mean distance, we want to estimate that. Well, second thing is let's zoom out a bit. Let's look at all the points. The observation here is that not all the points are equally important. For example, this guy and this guy, they are closer to the midpoint, right? So I really need to estimate them really accurately. But for those guys, as long as I obtain a few samples, I will have a pretty good idea, those guys are not going to be the midpoint. So which says that probably we can do some adaptive estimation here. And for each of the points here, I will give it a different confidence interval of different width, right? So in this case, I'm investing my computing power mostly on those points that are closer to the middle. But those guys, I'm spending minimum amount of power, computing power. And by doing that, potentially, I can save a lot of computational cost. Well, what is the desired goal here? Desired goal is to make something like this happen. And what I mean by that, the upper confidence bound of the best guy is smaller than the lower confidence bound of the second smallest guy. And if this thing happens, then we can say with high probability, this guy is the middle. Well, great. So 2 things. One is converting a computational problem to a statistical estimation problem. Second is that we can do adaptive estimation. Well, the third question is that how do we exactly do the adaptive estimation here? Well, so this adaptive estimation has been studied, very thoroughly in the community of multi-element bandits. So the idea for multi-element bandits is as follows. So suppose we are in a casino and there are a lot of vending machines. And I'm showing you one machine here. And it has a mini reward of the machine. So, and I don't know that ahead of time. So what I can do is that I can pull that machine sometimes. And those give me some observations. And then by looking at those observations, I can have a rough estimate of the mean reward. The actual multi-unbounded setting, it deals not only with one machine, but with a lot of machines. Suppose we have n machines. And for each of the machine, it has a different mean reward. So which means that some machines are better than other machines, and you would like to pull them more. So then it naturally gives you an adaptive estimation problem. And specifically the setting is that, so it's a game that for rounds t equals 1, 2, 3, every round you select an arm to pull. And by doing that, you can get a reward from that arm. And based on the observations, you need to figure out, which machine is better than other machines. And well, the trade-off here is pretty apparent, right? So some machines, after you pull 2 or 3 times, you know that they are apparently pretty bad. So I will not pull them anymore. But some of other machines that are closer to the best machine, but I need to pull them a lot of times to make sure that they are just slightly worse than the best machine. And specifically, one problem in multi-unbounded is called the best arm identification problem. And as its name suggests, its goal is to select the best arm. And the criterion here is that you need to select it first with high probability. And second, what you want to optimize is that you want to minimize the total number of arm pulls. Great. Well, this thing is a lot like the middle problem I have just introduced. Why? So it seems that there is a one-on-one correspondence of the two problems. The first thing to notice is that we can view every point in the middle problem as the arm. And then what is the arm parameter? The arm parameter, the mean rewards, is the mean distance for every point that we want to estimate. And then, so by pulling an arm, I'm actually obtaining an actual distance sample for that point. And with more distance samples, I can have a better estimation of its mean distance. And what is the best arm in the context of the middoid? That is the middoid. Well, the middoid is the point with the smallest mean distance, which is kind of a flipping of the arm with the biggest, the largest reward. That's fine. It's symmetric, so it's not a problem here. Which says that the middoid computation problem can be casted as an exact best arm identification problem. And which also says that if this is true, then we can just use any best arm identification problem algorithm and run it on middoid and see what happens. And we did exactly that. So here is the algorithm that we developed, which is based on the UCB type algorithm from the best arm identification problems. And so for the initialization part, step that we require certain samples for every point to have an initialized confidence interval. And the rule here is that every time I pick up the one with the lowest, the smallest, the lowest lower bound to pull, and I acquire another sample for that arm. And I keep doing that. And now it's this guy, and now it's this guy, and I keep doing that. And I keep doing this until a point where the upper confidence bound of the best arm is smaller than the lower confidence bound of the second best arm. And I stop here. And that is the middle point. Okay? So another question is, it seems pretty good that we didn't even implement our algorithm, which is not true, we actually implement that. But it's pretty good that we can just directly borrow an algorithm from another community to solve a problem here. So what is the computational complexity here? So there are some assumptions, and you can ask me later if you are curious, but it's basically n log n on real data. So, which means that, so theoretically, it can be improved to n log n, which is tight, but it's almost linear here. Okay?
So I have something I understand. Suppose that your matrix contains all ones, except one entry that is 0.
And then you will have this case. So those are my assumptions here.
So what is your assumption?
There are two assumptions here. The first assumption is that, so all the elements along this column, they are like homogeneous. We assume that if I sample without weight replacement from this column, that follows a sub-Gaussian distribution. And the second part, in order to derive this point, I need a distribution on all the mean distances. I have some figures at the end of the talk, and so if the mean distances, or the distribution of the mean distances follows some bell-shaped curve, no matter it's Gaussian or exponential, it's fine. It's a slightly heavier tail, it's fine. It gives you n log n. But if the distribution here follows a uniform distribution, then it came back to n to the 3/2, this number. But still, the dependency on the dimension is linear. It's not exponential. Great. So, what have we learned here? We have learned two things. One thing is that instead of computing the mean distance for each point exactly, we would like to estimate them. And second is that not all points are the same. Some are more important than others. So we can adaptively estimate them. And this is done using multi-embeddedness. So this is the first part of the talk. And hopefully you have understood this part. So let's move on to the second part. Well, so middle is nice. And then after this work, we are pretty happy with that. And we were thinking, how it can be used in some other scenarios, because it seems that the idea here is pretty general. So then, last summer I did an internship in a company, and where I need to use a Monte Carlo permutation test to compute the p-value for a lot of testing statistics, and that was terrible. And usually, so I need to compute it for 500 times 20, it's like 10,000 p-values, and each of them I run the Monte Carlo test for 5,000 times, and takes 20 minutes. But the resolution there is pretty bad, and if I want a better resolution, I need to go further, and then that takes forever. So then at that time, I was like, I don't really know what to do with that, and it's only after I came back to Stanford, and then I was like, I want a new project, and it seems that this problem is important because I have encountered it during my internship. So can I do something better than computing the same number of Monte Carlo simulations for every hypothesis, for every p-value? And so this is this work, adapting Monte Carlo multiple testing. Well, so as I mentioned, it deals with the context, which takes a bit of a long time to build. First, I will talk about hypothesis testing. Second, the multiple hypothesis testing. And third one, what is the Monte Carlo permutation test? And here you go. So the context is a large-scale multiple testing that we have an hypothesis test. And just give you a bit of a intuition here. Suppose you are trying to understand what the relationship between human genome and Parkinson's disease. We know that human genome, the variation across the human genome can be represented by a set of SNPs. And SNPs means the single nucleotide polymorphism, or the single point mutations on human genome. And there are like roughly 5 million is the right number for you to have an idea. So, and basically the hypothesis test, the ith hypothesis test is that is SNP I related to Parkinson's disease. Okay? So, and for the hypothesis test, and then we need to make a decision for each of SNPs to say they are related, they are not related. Okay? So then with the hypothesis test, we will collect some data and we will compute a p-value for each of the hypotheses. So the ideal p-value, the definition of the ideal p-value is the probability that your non-statistic, the statistic that you generated from the null model where the SNP is not related to the Parkinson's disease. That thing is more extreme than your observed statistic. But this logic is a bit convoluted, and so here it goes. If the non-statistic, if the p-value is small, it means that under the null model where the SNP is not related, then it's unlikely to observe your data, which means that we have a very less evidence for the null model where SNP is unrelated to the disease. which in turn supports that maybe the alternative hypothesis where the SNP is related to the disease is more likely. So, but it doesn't prove that, but it still gives you some evidence that the SNP is related to the disease here. Great. But the problem is that in settings like, for example, in settings like genome scale testing, usually we don't have a very good assumption for the null model, for the null distribution. But in these cases, here's the setting for the study I just talked about. Basically, what you do is that you find a bunch of people, and those are the people who have the disease, and those are the people who don't have the disease, and you measure their genotype, which is a discrete number. And you want to test the relationship between the disease and the genotype here. So in some cases, we don't have the null model, or our assumptions may suffer from model mismatch. But in those cases, often the simulation-based methods are easier, that they don't suffer from model mismatch. And so specifically, you can obtain n Monte Carlo samples of the null statistics. And here is a bit of idea that why this is true, why it's hard to know the distribution, but it is easier to have samples from the distribution. For example, if we are going to test the correlation here, what I can do here is that I can randomly permute the genotype or phenotype. And because I randomly permute the genotype, which is not dependent on the phenotype. So by definition, the correlation between the phenotype and the permuted genotype is from the null model, that the two things are not correlated. So in this case, we can generate the samples from the null distribution. And it is a gold standard. Well, because it requires only minimum assumptions of the distribution that under the if the two things are not correlated, all the samples are permutable. So the p-value usually derived from this study is usually valid. Great. And the Monte Carlo p-value is just an average of n Monte Carlo comparisons. And this guy is the indicator function. And this guy says that this guy is 1 if the null statistic is greater than the observed statistic and 0 otherwise. And you just take an average of all the things. And this 1 here is just to make the p-value exact, meaning that it's stochastically greater than the uniform distribution. But that's not important here. Great. So now we have the Monte Carlo test. And the second thing is that from the p-values, we need to derive decisions here, which is a subset of SNPs that we call significant. And one of the most popular way to do that is called the Benjamin-Hochberg method. And this is really popular. And by the way, this number was from 2 months back. And last yesterday I checked that, and this number became 54,000 citations. So it's really it's really hard to keep track of that. So what it does is that we want to control a quantity called FDR, or the false discovery rate, which is the expected value of the proportion of the false positives. And it only control it under the level alpha here. And what it outputs, its input is the set of p-values, and its output is a threshold and tau star, which is data dependent. And then it will select all the SNPs if their p-values are smaller than tau star here. Now here's a bit of intuition for those of you who do not know multiple hypothesis testing. Here's an example that why do we need a data-dependent threshold instead of a data-independent threshold? What if I select all the SNPs if their p-values are smaller than 0.05? What will happen here? Well, what will happen here is that most hypotheses are null hypotheses, and under the null hypothesis, p-value are uniformly distributed, which means that the number of false positives is roughly 5% of all the hypotheses. So the number of false positives is a number which we have a pretty good idea of what this number is going to be. But the problem is that different data sets have different strengths of their signals. Some data sets they have more discoveries, and some data sets they have less discoveries. So the number of discoveries here is really a data-dependent quantity. But the proportion of false positives is a ratio between the two, and the numerator is we have a good idea, and the denominator is data dependent. It means that FDR it can be either too large or too small. So we really need a threshold that is adaptive to the data set. Okay. Well, so far so good. So here's the procedure. And that procedure is very simple. First, I have this set of p-values, and I order them from the smallest one to the largest one here. And then I draw a line. And the slope of the line, the ranked case here, the slope of the line is alpha over m. And the procedure says that you find the guy, you find the largest p-value, who is below this line, and you select all the things below that. And that is the procedure here. And so K star is the number of discoveries, and tau star is the corresponding BH threshold, and we have this nice relationship here. And basically, tau star is the p-value of this guy. Okay. Well, so far so good. And so we have a pipeline where always we can have a valid p-value, and we have a multiple hypothesis testing procedure that is mostly widely used. And what's the problem here? The problem here is the challenges from computation. So here is a very small GWAS data set, that the genome scale multiple testing data set. So it has this data set is from 13 years ago, from 2006, and it has 500,000 of hypotheses. And usually the number of Monte Carlo simulations we run for every hypothesis should be a few orders of magnitudes larger than the number of hypotheses here. So I chose it to be 100 times larger than m here. And that gives us this many of Monte Carlo samples to compute. And so we have a great server in our lab, thanks to David, which has 64 cores. But that takes 2 months to compute this thing. And not to mentioning, this is not the state-of-the-art data set. The state-of-the-art data set, this guy will be 10 times more, 5 million. And this guy, and also there's another dimension, is the dimension of the number of peoples. And here the number of people is 500, and it can scale all the way up to 100,000. So no, people are not going to use it. Well, in their recommendation, they are going to use it. They are not going to use it unless necessary. Usually, one way to do it is that first you compute the p-values using some theoretical methods, and then you grab all the guys with small p-values and run Monte Carlo test on the small p-values. But there you have a problem of a selective inference because you have used the data sets twice. So yeah. So the point is that it's not people don't like to use it; it's that people cannot use it. And then question here: can it be made faster? Well, so here is a brief overview of our results here. So the first thing is that we have an algorithm called AMT, adaptive Monte Carlo multiple testing. Reduces the time, this is the workload time, and it reduces time from 2 months to an hour to compute. And so here is so this one is on the real data set, and this is for simulation. And the blue curve is the running time for the standard pipeline, where I'm showing you here x-axis is the number of hypothesis tests, and y-axis is the average number of Monte Carlo samples per hypothesis, and which is n here because we run n Monte Carlo permutations for each of the n tests here. And here is the performance of our algorithm, and so. If we just do a linear regression here, and we'll see that this is a log scale, of course. We'll see that the dependency on n is roughly square root n. And so And then let's prove some theorem on that. So here's the theorem. The first is a correctness that it always guarantees the same discoveries as the standard method with high probability. And in practice, this probability, the failure probability is something like 1/1,000,000. So yeah, this is not almost negligible. And the second is the running time. And so the expected number of Monte Carlo samples it takes is square root n times n, which is almost exact as as the simulation suggested. And the third one is the optimality. And first, I need to explain what do I mean by a correct algorithm. A correct algorithm, what I mean is that I always return the same discoveries as the standard pipeline with high probability. And it can be shown that for any correct algorithm, it takes at least this much of samples. And
Does it have a constant?
yeah, there's a constant there. Yeah, so actually O tilde because there is a log factor there. Great. So core idea. And what I'm going to do is I'm going to repeat all the slides that I had before again. So let's focus on the first hypothesis. And those are the n Monte Carlo samples for the first hypothesis. Okay. And as a reminder, the p-value here is an average of these n comparisons here, and which means that each of this guy is a Bernoulli random variable, and whose average is almost the same as p1. Then we can do estimation with a smaller number of samples, right? So we can have a p hat here, and we know that the accuracy is roughly proportional to 1/square root of n1, the number of samples that I use. So this is the first step from computation to estimation. Well, second step is that how do we do adaptive estimation? Well, so in order to recover the discoveries by the standard pipeline, I need two things. One thing is that I need to estimate the BH threshold, which, as I mentioned before, is data dependent. And second is that I need to know how each of the p-value compares with the threshold here. And you see, it already suggests a scheme for adaptive estimation because those guys are far away from the threshold, and those guys are closer to the threshold. So we can estimate them with different accuracies. And then let's bring back the old picture of bottom bandits. So one thing to change here is that previously we considered the best arm identification problem, but here, because I'm selecting a subset of hypotheses, and you know that we can treat every hypothesis as an arm. So we are selecting a subset of arms. So let me change it to best k-arm identification, which is another line of work. And the goal here is to select the best k-arms. Okay? And so in your study, the k is a given number, the best 2 arms, best 3 arms, and so on and so forth. And now we have this picture, multi-arm bandits, and the adaptive and Monte Carlo multiple testing. And the arms corresponds to the hypothesis, and the mean reward corresponds to the Monte Carlo p-values that we computed by averaging all n Monte Carlo samples. And the arm pools corresponds to obtaining 1 Monte Carlo sample for one hypothesis. Well, one good thing here is that previously I have seen the sub-Gaussian for the sub-Gaussianality for the samples, but here I don't need that because those samples are Bernoulli. So which means I can have a much tighter confidence bound for those estimates. And so, too much things. And so the best k-arm corresponds to the BH discoveries. But there is a caveat here. The caveat here is that for the best k-arm problem, k is a given number. So if I were to just use the best k-arm identification algorithm, I need to use k as an input. So what is k? Well, if you go back, you'll see that those are the number of discoveries. So k is the k star here. And this is really bad that k star, this quantity number of discoveries, is data dependent. So I'm not able to know it ahead of time. So which means that I cannot just directly use a best k-arm identification algorithm for my purpose. And to be more specific, the problem of Monte Carlo multiple testing is more like a problem of a best k-arm identification problem where k is a data-dependent quantity and is determined by the Benjamin-Hochberg procedure. Which means that I need a new algorithm for this case. Well, but I can still borrow, all the, high-level ideas from the multi-unbounded community to design our algorithm. And specifically, it goes as follows. So first, the thing to know is that you need to maintain two sets of estimates. One is for the p-values, and the other is for k, for the number of discoveries, or for the BH threshold, because there is a nice coupling between the two that tau equals k over m times alpha. So maintaining one is the same as maintaining the other. And as the initialization step, I would initialize k to be the biggest number possible. And the best case is that we reject all m hypotheses. So I start with m here. And correspondingly, tau star is a relationship equals alpha here. And the idea is that I adaptively move this threshold down until I reach the true threshold. And on the same time, I would like to maintain an upper and lower confidence bound for every p-value. And because now I don't have any samples, so the upper bound is 1 and the lower bound is 0 for all the p-values here. And the third thing is that every time I'm going to sample a batch of Monte Carlo samples instead of one. And I'm going to sample a Monte Carlo sample for a hypothesis if its p-value is uncertain. And the definition of uncertain hypothesis is pretty straightforward here. So if its confidence bound crosses this threshold, it means that we are not sure if the actual p-value is smaller or bigger than the threshold. So I call it uncertain. And here the rule is to sample uncertain hypothesis. And one thing to mention here that I'm going to use in the next slide is that because I apply this top-down procedure that I keep decreasing the threshold as long as I'm certain that the current threshold is not going to be the true threshold. Then I always have the relationship that our current estimate of threshold is greater or equal than the actual threshold. And I'm going to use it in the next slide. So the first thing, the progression of the algorithms is that when we keep sampling for each of the hypotheses, their confidence bound become narrower. And then we can, for example, this guy, its lower bound is greater than the threshold, so it becomes certain. So I no longer sample it. So this is the update for the p-values. And second is the update for the BH threshold. And here is a bit of logic here that requires a little bit of attention. So let's look at this guy, P3. So because it's a lower confidence bound, it's greater than the threshold, then we know that with high probability, P3 is greater than the threshold. And because our current estimate of threshold is greater than the actual threshold, this means that P3 is greater than the actual threshold, which also means that the third hypothesis will not be selected at the end. Okay? And because K star is the total number of discoveries I'm going to have at the end, and the P3 is not going to be one of them. So I know that K star is at most I minus 1. And that is a confirmation that I can further reduce the estimate of the BH threshold here by 1 here. And I keep doing that. And I still keep sampling the uncertain hypothesis here. And I keep doing that until I reach a point that all the guys are either above or below the threshold for certain. And basically, the theorem says that if that happens, then we are good. But those guys correspond to the discoveries from the standard pipeline, and that's the algorithm. Well, so what we learned from that? Well, we learned that instead of computing the p-values for each test exactly, let's estimate them. And adaptive estimation using multi-unbanding. This is basically the same slide as before. Well, so this is mostly almost all of my technical part of the talk today. And a mentioning here is that these two work, the first and the third word, are my works, and they are not the only works along this line. And there's another work by nice work by some other colleagues in our lab. And when we started the first work 2 years ago, and it seems that this idea is pretty general, and I do encourage you to apply it to situations you find fit. And so, and then it's an overview of my PhD works, and I wouldn't bore you with the details, but so there are basically 3 things here, and today I talk about the algorithm acceleration, and I'm also pretty obsessed with the idea of the data integration. And the idea is like currently you have a data set, and because the size of the data set is limited, so it's not powerful enough. But what you can do is that you can grab some publicly available data sets from other places which are relevant but not exactly the same as your data set. Because if they're the same, you can just combine them. And then you can think of all sorts of interesting methods to combine the 2 data sets which have different modalities to either increase the power or broaden the scope of your investigation. And we have done this in the context of multiple testing and with dimensionality reduction and classification. So, and then the third one is data collection. Is that if you are the guy who is going to collect the data, then you can think of how to design your experiments. And we did it specifically for the single-cell RNA-seq, and we identified the optimal trade-off and the optimal estimator there for the experiments. Well, so, and here comes the part for the acknowledgement. Right, so first is the acknowledgement for my funding sources, and so I was very lucky that I got quite a few fellowships. So, which are nice, which means I don't need to do a lot of TAs. And also I would like to acknowledge NSF and NIH for their support. And the second part is committee members. And first I would like to thank you guys very much that I have changed my hour difference time quite a few times, and I'm really appreciated that you guys can accommodate that. And so I guess my connection with Andrea is that when I do theory, there are always 2 lecture notes that I would like to keep in my computer. One is Andrea's note of EU 278, and the other is Zhang Duqi's note of stats 311. And if you haven't done that, do that. They are very useful. And so I first came here, we explored the idea of empirical Bayes, which is quite relevant. It's not apparent first, but which is quite relevant to the work that Saki has done with Jian Tao and Yan Jun on polynomial approximation and functional estimation of discrete data sets. So I have read all your papers on that topic, which is a law paper.
But are they good?
I like them, and I have studied them a lot. And Trevor. So the first time I met Trevor is last year. I took one of Trevor's classes, that's 305C. And then after the class, every time I met Trevor, I always told Trevor that this class is the best class I have ever taken. And I keep telling him up to a point that he said, now you're flattering. But yeah, I think that course is very nice. Yeah, and I think I have read some of your papers, and my favorite is not the Lasso paper, but the paper on gap statistic. And the reason is that, I have done 2 internships, and for the first internship, we need to cluster something really carefully. And surprisingly, the gap statistics works there perfectly, so which is nice. And here are my mentors. They are not officially my advisor, but they mean a lot to me. And the first is Fabian, Professor Fabian Peace, and Professor Junior, who is now here. And my relation to them is that before I came to Stanford, I did a summer internship here in the year of 2013. And that time they were my mentors, and I think they taught me a lot of how to do research and how to be passionate about things that you're interested in. And after that, they were sort of more like my life advisor on whenever I make some decisions. And in fact, the decision that I decided to work with David, I also consulted them a lot, and they were like, you're a nice guy, so.
Thank you.
Yeah. Another thing I learned from them is that Fabian always told me that the fact that you have a lot of fellowships doesn't mean you're going to produce enough researches, and he has saw a lot of cases where the student has SGF and sort of wasted the first 3 years of their life, and because they were like, I'm not in a hurry. So during my PhD, I'm always in a hurry, and always try to work hard and try to produce something interesting. And the third one is Professor Alan Oliski from University of San Diego. University of California, San Diego. So when I first met with David, he introduced me to Alan, and the three of us have worked on things on the polynomial approximation and empirical Bayes. And yeah, that was quite nice because when I first came here, I'm a PhD knowing nothing, young and naive, and both of them are very senior, but they are super patient and really supportive. And yeah, and what I learned from Alan is that information theory is pretty hard. So I sort of switched to computational biology. So yeah, so and I met James in the third year of my PhD, and then we switched to more on the applied territory of the entire landscape of the research. And yeah, I think I learned most of the computational biology from James, and apparently we have a lot of papers, and I thank you very much for that. And yeah, I think what I learned is I know how to be an applied statistician and to use the techniques effectively and efficiently without worrying too much about the theoretical details, and only care about those things that matters. Which works in computational biology. Okay, so I'll stop here before talking about too much things.
Stop now.
And finally, my advisor David, and yeah, I think yeah, apparently along this year, so we came a long way from to here, and well, what to say? So I guess the best thing I learned from you is that how to develop a good taste of the problem, to identify the problems that will have a high impact, that are useful in practice, but still have a theoretical value, and not to be bothered about the details of the proofs, but to show the main ideas there, that matter. So I think it's a nice combination of applied science and theory. And yeah, I think that the taste of the research is the best thing I learned from you. And of course, all these years you have given me a lot of support, and I always appreciate that. So yeah, and other guys, my collaborators, and some of you are here and some of you are not here. And also my lab mates here. And so there's a new guy, Luis, who's going to come to our lab this summer. I haven't put your name here, sorry. And so those two guys are changing from my lab mates to my collaborators. So I put their photos here. So my lab mates, if you want to move from here to here, we still have some time. Yeah, so so I have done 2 internships, one at Genesis and the other at Grail. And also, the first is on the next generation DNA sequencer, and second is on the more on the downstream analysis of early cancer detection. And that was a lot of fun. And I have drawn a lot of inspirations from these researchers there for my research at Stanford. And so my friends here, and so a special mentioning here is that Yuzhou, who is really kind and made all this nice cake here and cookies. Those are outside. If you haven't tried that, make sure to finish them. And yeah, and also, well, it has been a lot of fun. And so this is my family, and my parents and my grandparents, and this is my girlfriend, and recently became my fiancée. So we're going to get married very soon, and this is my dog.
Is that a cat or something?
It's a corgi. He's really fat.
Yeah, also.
I also have a cat, but yeah, she passed away a few years ago. So yeah, she's pretty old. And so one fun fact is that both my parents and my fiancée, they are doctors, and so when I was in high school, I was like, no, I'm not going to be a doctor. And then I studied physics, and I studied EE at Tsinghua, and came here to do in EE department. And now it seems that I'm slowly switching, from physics to engineering and to biomedical data science. So I don't know, it's like karma, and that's karma it is. And that's all. Thank you for the day.
Great. So, any quick question for Martin from the public part of the talk? There will be more questions afterwards. Don't worry. Okay. Great. All right. So let's take 5 minutes break.
Great.
And then we will proceed with the close part.